Hi guys, Miss Francis here. We left off discussing Thomas Hunt Morgan's discovery of of genes that are linked to the sex chromosomes. He used fruit flies to do his experiments where he learned that eye color is linked to the sex chromosome. Now we can represent and explore these chromosomes. We could explore the offspring that would come from parents that have these genes that are linked to their sex chromosomes, doing Punnett squares. Sorry, doing Punnett squares and pedigree charts. So let's start out with a Punnett square. So the nomenclature for sex-linked Punnett squares is similar to the regular nomenclature, except that we're going to use the sex chromosomes with a superscript. So the sex chromosomes for the female are XX, and the sex chromosomes for the male is XY. So if we're talking about a female, right? If we're talking about a female, we're going to use XX. And we use the same nomenclature, capital letter, lowercase letter, to represent something that's dominant versus something that's recessive. So color blindness, being a hemophiliac, baldness are all recessive diseases that are sex-linked. So we're going to let the capital N represent a normal individual, and a lowercase n represent an individual that has one of those sex-linked diseases. Now check out the possible genotypes of the female. The female can be either homozygous normal or heterozygous normal and still not have the disease. However, she can have the disease if she has the recessive alleles from both parents. Now, if we're talking about the male, if the male has a normal genotype, it would be XY with the capital N. But if he has the disease, he only needs one copy of the recessive allele in order to get the disease. So this is why these sex-linked diseases are more common in men than in women, because he only needs one dose, so to speak, of the gene to either be normal or to have the disease. So let's explore what Thomas Hunt Morgan did with his fruit flies. So let's use our nomenclature over the nomenclature that's here. So if we have a female and male flies, and we have a P generation of true breeding red and true breeding white. Well, I'm going to use the nomenclature X, capital R, X, capital R for the female. In other words, capital R is red, lowercase r is white. So then the nomenclature for the male would be X lowercase r Y, because he's true breeding blue. If we are to look at the F1 generation, do you notice that all the females and all the males will be heterozygous, but they would express red eyes? And that follows Mendel's pattern of inheritance of dominance. Now, if we take those F1 generation, those heterozygous flies, and we look at the F2 generation, their offspring, do you notice that I have that 3-to-1 ratio, right? I've got 3 flies with red eyes and 1 fly that has blue eyes. Now take a look at who has white eyes. Who has the white eyes? The white eyes are only found in the male, whereas the red eyes can be found in either the female, so these two guys are females that have red eyes, or can be found in the male. So that's how Thomas Hunt Morgan knew that something besides for dominance, which is shown here, was going on. That even though he still got the 3-to-1 ratio, that ratio was off. No, it's not off. It's just that one, that white-eyed, was only expressed in the male. So that's how he knew something was going on. So let's try a practice problem. It says, in humans, colorblindness and hemophilia are sex-linked recessive disorder. Cross a colorblind female, so we've got a female, with a male that has normal vision. So here's our male. Now, I'm going to use the letter N, capital N, for normal, and I'm going to use lowercase n for colorblind. In order for a female to be colorblind, she needs to have both colorblind alleles on her chromosomes. So she needs X lowercase n, X lowercase n. So that would be the phenotype of the female. Now, in order for our male to be normal, he would have to be capital N on his X chromosome, because if he had the lowercase n on his chromosome, well, then he would be colorblind. So if we do this cross, I'm going to put mom on the top and I'm going to put dad on the side. And when I do the cross, I get X capital N, X lowercase n, X lowercase n, Y, X capital N, X lowercase n, X lowercase n, Y. So let's look at the children overall. So overall, 50% of the children will be carriers, and those carriers are all females. They're going to be normal, however. If they're talking exclusively about females, well, then we would say 100% of our females are normal. If they are talking exclusively about the males, if we look at just the males now, 100% of those males are going to be colorblind. But overall, the children in totality, only 50% of the children are going to be normal. Right, so let's look at another practice problem. A hemophiliac male with a female that's not hemophiliac. So if I go to a hemophiliac male, so I'm going to say capital H is normal and lowercase h is hemophilia. And if I cross a hemophiliac male with a female that's not a hemophiliac, but whose father was, well, that means that she would have to be a carrier. So let's do this cross, right? So I draw my box for my Punnett square. I'm going to put dad up here, and then I'll put mom along the side. And I would get X capital H, X lowercase h, X capital H, Y, X lowercase h, X lowercase h, X lowercase h, Y. So if I look at this cross, I can see if I separate my females from my males, right? These are my females. So one of my females has hemophilia, the other female does not. And if I were to look at just my males, again, one male will be hemophiliac and one will not. All right, and here's another practice problem that you guys could do. I would recommend also doing the practice problems in our Mendelian genetic note packet. So let's take a look at what a pedigree chart might look like that shows a sex-linked trait. Pedigree charts are like puzzles sometimes. So this pedigree chart is showing an X-linked recessive trait. Well, how do I decipher this? It's easy to decipher something as being recessive versus dominant. It's harder to tell if it is X-linked, Y-linked, or autosomal. So here we know that it's recessive because I look at these parents and they're not affected, but they have a child who is affected. That means it's got to be recessive. Now, to know that it's X-linked over autosomal, it means more males than females are affected. It's never passed from father to son, and all daughters of affected fathers are carriers. So now moving on, how would I decipher a pedigree chart showing an X-linked dominant trait? Well, we know that dominant traits are present in every single generation. We know that just because one of the parents have it doesn't necessarily mean that all of the offspring are going to have it. But now, how do we know whether it's sex-linked or autosomal? Well, both males and females will be affected. Often it's more females than males. The affected sons must have an affected mother. The affected daughters can have either an affected mother or an affected father. And the affected fathers are going to pass on that trait to all of their daughters. Looking at a Y-linked inheritance pedigree chart is much easier. Do you notice that all of the boxes that are colored in are boxes? They're not circles. That means it must be Y-linked because it's only affecting the males. Sometimes it's not as cut and dry, and you need to kind of go back in the pedigree chart and see if you could figure out the genotypes of the different generations in order to complete the puzzle, so to speak. In other words, to tell whether or not we're dealing with an autosomal trait or a sex-linked trait.